Have you ever paused to ponder the intricate workings of our body, specifically how it converts glucose into energy? It's a fascinating process, a dance of molecules and enzymes known as glycolysis. Today we delve into this biochemical ballet, breaking down each step of the process. Glycolysis is a 10-step metabolic pathway that occurs in the cytoplasm of our cells. It begins with a 6-carbon molecule of glucose. Through a series of reactions, this glucose molecule is skillfully transformed into 2 3-carbon molecules of pyruvate. The first step is called the investment phase. Here, glucose is transformed into glucose 6-phosphate by the enzyme hexokinase. This reaction requires the energy of 1 molecule of ATP, which is why it's called an investment. Step 2 sees glucose 6-phosphate rearranged into its isomer, fructose 6-phosphate, by the enzyme phosphoglucose isomerase. The isomerization process prepares the molecule for the next phosphorylation. In the third step, phosphofructokinase uses another ATP molecule to convert fructose 6-phosphate to fructose 1,6-bisphosphate. This step represents a key point of regulation in the glycolysis pathway. The fourth step involves the splitting of fructose 1,6-bisphosphate into 2 3-carbon sugars, dihydroxyacetone phosphate and glyceraldehyde 3-phosphate by the enzyme aldolase. The fifth step is a swift interchange. The enzyme triose phosphate isomerase quickly converts the dihydroxyacetone phosphate into another molecule of glyceraldehyde 3-phosphate. Now begins the payoff phase. Here, the cell starts to see a return on its initial energy investment. In the sixth step, each glyceraldehyde 3-phosphate is oxidized to 1,3-bisphosphoglycerate by the enzyme glyceraldehyde 3-phosphate dehydrogenase. In this process, 2 NADH molecules are formed, capturing some of the energy. Step 7 involves the production of ATP. The enzyme phosphoglycerate kinase transfers a phosphate group from 1,3-bisphosphoglycerate to ADP, forming ATP and 3-phosphoglycerate. In the 8th step, the enzyme phosphoglycerate mutase rearranges 3-phosphoglycerate to 2-phosphoglycerate. Step 9 involves the enzyme enolase, converting 2-phosphoglycerate into phosphoenol pyruvate, releasing a water molecule. Finally, in the 10th step, pyruvate kinase transfers a phosphate group from phosphoenol pyruvate to ADP, forming another ATP and the final product, pyruvate. In summary, glycolysis is a series of 10 reactions that convert 1 glucose molecule into 2 pyruvate molecules, releasing energy in the form of 4 ATP molecules and 2 NADH molecules. This process is fundamental to life, providing the energy that fuels our cells. So the next time you savor a sweet treat, remember the intricate dance of glycolysis, the molecular ballet that transforms glucose into the energy that powers life.